this video, we are going to be looking at DNA replication. As I described in a previous video, that replication is making an exact copy of something. DNA replication is when DNA makes an identical copy of itself, exactly the same. It occurs during interphase. Interphase is the phase that is a preparatory phase that occurs before any type of cell division, whether it is mitosis or meiosis. The cell has to prepare itself by replicating its DNA so that there is more genetic information. So this DNA replication occurs during interphase. Up next, we're going to talk about the significance of DNA replication. The significance of DNA replication is to ensure that each daughter cell has identical DNA composition to the mother cell. This is the mother cell. What this means is that this mother cell undergoes DNA replication so that its daughter cells at the end of this cell division have an identical DNA composition, so that their genetic material is identical to that of the mother. That's basically what it means. Yeah, even if you don't understand what it means, just know the answer. To ensure that each daughter cell has identical DNA composition to the mother cell. Yes. So getting into the real process. This is from a marking guideline. It's best you stick to the way it's giving the way the process is given in an exam guideline. This is most of the time it's a 6-mark question when they tell you to describe the process of DNA replication. It's best to stick to the standard that they want. Don't be creative and go look at textbooks. Most of the time textbooks don't really give what the marking guideline wants. It's only specific and very few textbooks that do that. So we are gonna start this process, DNA replication. The first one is that the double helix DNA unwinds. Unwinding, it basically means it untwists. You see what is happening here? This is it untwisting / unwinding. The double-stranded DNA unzips. This is it unzipping. You see, when a zip opens up, it looks very similar to what is happening right here. The process of the double-stranded DNA unzipping is basically the weak hydrogen bonds breaking to form two separate strands. You see, after the unzipping, two separate strands form. Both strands are used as templates. By template, what is meant is that both of them act as, I'd like to say, they allow a complementary strand to form. Like they're basically the base of the complementary strands forming. Yeah, something like that. Yeah. And then to form complementary strands, like I'm saying, complementary strands, it basically means the nitrogenous bases complement each other. They follow base pairing. You see right here, it's A and T, T and A, just like that. Same thing this side, C and G, T and A. That's what complementary strand means. The complementary strand also connects. It bonds to the original strand following base pairing. That's basically what it means. Just some fancy words that they're trying to confuse you, but yeah, it's not that deep. And then using free floating nucleotides from the nucleoplasm. I want you to understand that all of this happens in the nucleus. It happens in the nucleus. So these nucleotides that you see here, this is a nucleotide, this is a nucleotide, this is a nucleotide. They are from the nucleoplasm. They are all from the nucleoplasm. Let's assume that this white area is the nucleoplasm. They are all from the nucleoplasm. So in the nucleoplasm, these nucleotides are allowed to freely float. So yeah, they are the ones that form the complementary strands. Yes. And then we come back to here we extend and say that adenine pairing with thymine. You don't really need to say that, but yeah, it's basically explaining base pairing. Two identical DNA molecules are formed. You see, this is a new DNA molecule, this is a new DNA molecule. They're exactly the same if you notice. You see here T, TA, TA, CG, CG, GC, GC. They're exactly the same. And then lastly, each molecule consists of one new strand and one original strand. You see, this is the original strand. This is a new strand, new strand. That's what that last point means. So that is DNA replication. Understand this, memorize it, cram it if you're gonna do that. But yeah, this is how you should know your DNA replication. That is it for today's video. Thank you. Let's get it.